Welcome to today's lesson on mitosis and the cell cycle. We will explore the essential stages of mitosis, understand the results of this process, and discuss how cancer arises from uncontrolled cell division. In our lesson today, you'll learn to identify and recognize the distinct stages of mitosis. We will also cover the outcomes of mitosis and emphasize how problems with this process can lead to cancer. So far, you have learned that eukaryotic cells contain DNA in the form of chromosomes within the nucleus. Additionally, you know that cell division occurs through the cell cycle, which includes mitosis. This foundational knowledge is crucial as we delve deeper into the specific mechanisms driving cell division. Advancing our understanding of mitosis enhances our comprehension of biological growth, development, and repair processes. Let's dive into the details of mitosis, a critical process of cell division that ensures growth, development, and tissue repair in multicellular organisms like humans. Mitosis is a process where a single cell divides to produce two new, genetically identical daughter cells. This process is essential for growth as organisms need to add new cells to grow. For repair, by replacing damaged or dead cells. And for maintenance, by replacing older cells with newer ones to maintain healthy tissue function. In humans, most cells contain 23 pairs of chromosomes, with one set from each parent, giving a total of 46 chromosomes. These chromosomes carry the genetic information necessary for all cellular functions. Recall that the cell cycle is a series of stages in the life cycle of a cell. Each cell goes through this cycle multiple times. Mitosis is a stage in the cell cycle. It occurs after the interphase and before cytokinesis. We can split the process of mitosis into four main stages: prophase, metaphase, anaphase, and telophase. As well as the cycle diagram we have on screen now, we can also represent the cell cycle in this linear format, which shows the interphase at the start, then the four stages of mitosis, then cytokinesis. Let's look at the four stages of mitosis in more detail. Firstly, let's look at the prophase. In this first stage of mitosis, the cell's DNA, which had been in a less organized form called chromatin, condenses into clearly visible chromosomes. Each chromosome appears as two sister chromatids joined at a central region called the centromere. The nuclear membrane, which encloses the nucleus, breaks down, allowing the chromosomes to be released into the cytoplasm. Spindle fibers begin to form from centrioles, structures that help in cell division, and move towards opposite poles of the cell. The second stage of mitosis is the metaphase. In this stage, the chromosomes align themselves along the middle of the cell in a single line. Then the spindle fibers attach to the centromere of each chromosome, readying them for separation. The third stage of mitosis is the anaphase. In this stage, the spindle fibers shorten, pulling the sister chromatids apart at the centromere. The separated chromatids, now individual chromosomes, are pulled to opposite poles of the cell. The final stage of mitosis is the telophase. In this stage, the chromosomes that have reached the poles begin to decondense back into their uncondensed form, chromatin. New nuclear membranes form around each set of chromosomes, creating two separate nuclei within the cell. After the final stage of mitosis comes cytokinesis. Some people classify this as part of the mitosis process, but we will consider it separately. During cytokinesis, the cytoplasm and cell membrane divides, forming two new cells. First, the cell's cytoplasm divides, resulting in two identical daughter cells, each with a complete set of chromosomes. Then the cell membrane reforms to separate the two new cells. So, at the end of mitosis, you have two daughter cells that are genetically identical to each other and to the original parent cell. Each daughter cell contains the full set of 46 chromosomes, 23 pairs, making them diploid cells. This ensures that the genetic information is completely passed on from one cell generation to the next when they divide. While controlled cell division is crucial for the body's growth and repair, uncontrolled cell division can lead to cancer. This often occurs when mutations cause the mechanisms that regulate the cell cycle to stop working. Cancer cells bypass the normal regulatory checkpoints of the cell cycle, leading to excessive and abnormal tissue growth and cause cells to divide uncontrollably, forming masses called tumours. These tumours can invade surrounding tissues and spread to other parts of the body through a process known as metastasis. Mitosis is a vital process that produces two genetically identical daughter cells from a single parent cell, ensuring growth, repair, and maintenance of tissues. However, when this process goes wrong, it can lead to uncontrolled cell division, resulting in cancer. Understanding the stages and regulation of mitosis not only highlights its importance, but also sheds light on how disruptions in this process can have serious health implications. By understanding the phases of mitosis, you can understand how genetic information is transferred and the potential risks of uncontrolled cell division, such as cancer. This foundational knowledge is essential for further studies in biology and understanding cellular processes in greater detail. Let's work through some questions to consolidate our knowledge on mitosis. Question 1. Look at the provided diagram. Identify the stage of mitosis where the chromosomes align in the center of the cell. We see that in one of the diagrams, the chromosomes are lined up at the equator of the cell. In the metaphase, chromosomes align in the center of the cell. So our answer is the metaphase. Let's try another question. Explain the result of mitosis in terms of the number of daughter cells produced and their genetic composition. Mitosis results in the production of two daughter cells. Each daughter cell is genetically identical to the parent cell and contains the same number of chromosomes. So, the result of mitosis is two genetically identical daughter cells, each with an identical chromosome count to the original parent cell. Great work. That's the end of our lesson on mitosis. Let's recap. First, we explored mitosis and its significance within the cell cycle. We started by recalling that mitosis is a type of cell division responsible for growth, development, and tissue repair in multicellular organisms. We outlined the main stages of mitosis, which include prophase, metaphase, anaphase, telophase, and then, following mitosis, we have cytokinesis. During prophase, the chromatin condenses into visible chromosomes, and the nuclear envelope breaks down. In metaphase, the chromosomes align in the middle of the cell. During anaphase, the sister chromatids are pulled apart to opposite ends of the cell. In telophase, the nuclear envelope reforms around each set of chromosomes, which then decondense back into chromatin. Following the mitosis stage, we discussed cytokinesis, the process that divides the cytoplasm, resulting in two genetically identical daughter cells. We highlighted that each daughter cell has the same number of chromosomes as the original parent cell, ensuring genetic consistency. We then moved on to discuss the importance of cell cycle regulation. Proper control mechanisms ensure that cells divide only when necessary. We examined how loss of these regulatory controls can lead to cancer, a consequence of uncontrolled cell division, which can lead to tumours and spread to other parts of the body. Overall, understanding mitosis and its regulation is essential for grasping how organisms grow, develop, and maintain their tissues, as well as comprehending the underlying causes of diseases like cancer. This knowledge provides a foundation for future studies on cell biology and disease mechanisms.
Watch the rest of the GCSE biology course at Sophos Education.